Yes,
Hi,
hi.
Juri.
Hello, everyone. I'm happy to share some knowledge about event-related potentials with you. So it's going to be introduction to event-related potentials. And my goal is to show to you what are the advantages of using event-related potentials in clinical practice, and what is the new information you can get from event-related potentials. And I start, of course, with Hans Berger. He was the first who recorded electrical signal from the brain. We call it electroencephalogram. And he was the first who found that human brain in the resting condition, in eyes closed condition, generate alpha rhythm. Sometimes we call them Berger rhythms. The rhythmicity about 10 hertz. Initially, he wanted to find a neuronal mechanism of telepathy, but actually it turned out that alpha rhythm, 10 hertz rhythm, is not enough to transmit the thought over the distance. And also he had a hope that EEG, electroencephalogram, that he discovered would be helpful for a diagnosis of different brain disorders. And basically, it never happened. All studies during the last years showed that the diagnostic value of spontaneous EEG is quite low. Especially when we are talking about adults. When we are talking about adults, like patients with schizophrenia, with OCD, with depression, and if you compute the grand average EEG spectra, you will get approximately the same curve. So there is no difference between different categories of patients, which actually tells you that the diagnostic value of EEG spectra is relatively low. Although in 1980s, we learned quite a lot about mechanism of generation of alpha rhythms, and in general, all types of rhythmicities. So the mechanisms are quite well known, but we have to keep in mind that spontaneous EEG reflect cortical mechanism of cortical self-regulation, and you wouldn't expect much from this. Okay, then here I show you the Berger waves and the recording using our system, using WinEEG software at the bottom. The next, 1964, Gray Walter, a famous English neurologist, and he was the first who discovered a CNV wave, the wave that he called contingent negative variation. He asked subject to do a task in which he presented warning stimulus, and a few seconds after he presenting the trigger stimulus, and he averaged the EEG fragment over trials, and he found that during the preparation to receive the second stimulus making button press, the brain generate negativity, which he called seeing V wave. And on the left, I show you his initial paper, the drawing from his paper. On the right, I show you the data from our studies. And basically, you can see that indeed the brain generates some centrally generated negativity. Basically, the mechanism of this negativity are not well understood. We still have a few ideas what is the mechanism of generations of seeing wave, but the diagnostic power of these waves was proven to be very high. For example, the CNV wave can be used for differentiating schizophrenic patients from healthy control. So there was quite a lot of research studies that showed that CNV wave is reduced, dramatically reduced in patients with schizophrenia. The next, approximately the same year, I think it's 1964, a group of scientists discovered P300 ERP wave. So they actually, and among them was Roy John, a neuroscientist from University in New York. And what they did, they actually asked the subject to perform the oddball task. So they presented the standard stimulus at about 1 Hertz frequency. And in some cases, instead of standard, they presented a deviant stimuli, and they asked the subject to press a button to deviant stimuli. So it's a kind of active oddball paradigm. And in this active oddball paradigm, they found that trigger stimulus, rare deviant stimuli, they generate the potential, positive potential that is distributed over parietal areas, and they call it P300 initially. Later on, they called P3B because they also discovered that some distractor stimuli generate a frontally distributed component, which they call P3A. Again, we don't know mechanism of generation of P300, although there are many papers with many theoretical views to this wave. But basically, we don't know the mechanism of generation of this wave, but the diagnostic power of this wave is quite high. As you know, it's quite often is used in devices like a lie detector. But it also was shown that latency of P300 is delayed in patients with dementia. And basically, P300 is a good neuromarker of dementia. So increase of the P300 latency is a good indicator of a subject having dementia. Okay, then 1978, Risto Natonen, a Finnish psychologist, discovered a small ERP wave, ERP component, which he called mismatch negativity. And he used a simple auditory oddball task, again, presentation of standard stimuli together with deviant stimuli. It was a kind of not active condition, because the subject has to read a book or to watch the TV program without sound. So actually it was a kind of passive condition. And in this passive condition, he observed that deviant stimuli, they generate negativity waves that is generated over frontal electrodes, and he suggested that this wave reflects some mechanism of change detection in the auditory cortex. And basically, again, the mechanism of this wave are not well understood. And together with Risto Natanen, we wrote a few papers, several papers, and the results from one of those papers are shown on the right. And we recorded event-related potential, intracranial event-related potential in oddball paradigm. We recorded these intercortical potentials in different parts of the auditory cortex. And as I said, the mechanisms are not known. There are at least two groups. One group say one thing regarding the mechanism of generation of mismatch negativity. The other group tells something different. And from time to time, one group is writing a paper, a theoretical paper, then Risto responds, etc., etc. And it's quite amazing that both groups, they cite this intercortical ERP paper that we published together with Risto, and one group think that it support its idea. The other group think that it supports their ideas. So as you can see, there is not much of consensus, and there is not much of understanding in terms of mechanism of generation of this rhythmicity, of this ERP component. But the diagnostic value of mismatch negativity is proven to be high. For example, it can be used for diagnosis patient with dyslexia. Okay, then here I have to mention Professor Bacteria. She was my supervisor, and she was the first who used intracortical, intersubcortical implanted electrodes, not only for diagnosis and therapy, but also for recording different physiological parameters from this electrode, including impulse activity of neurons, intercortical potentials, the oxygen level of oxygen, etc., etc. So, and I think that our group was the first who discovered that the basal ganglia are involved in cognitive function. Now, if you open any neuroscience book regarding basal ganglia, you will find that it's well accepted that indeed the basal ganglia are involved in cognitive function, but in 1970s, 80s, it was not people, they thought that the basal ganglia are involved mostly in motor functions, not in cognitive function. Well, anyway, that is then here I show you the sources of electrical fields that can be recorded from the scalp. So as you know, in the cortex, we have different type of excitatory and inhibitory cells. And those cells are the source of electrical fields on the scalp. One important fact is that the membrane, the neuronal membrane, is electrically charged. It has ion channels, and ion channels can be open or closed due to the dynamics of neurotransmitters and also potential. So as you know, there are ligand gated channel and voltage gated channel. So this ion channel, they produce different types of currents, and these currents that flow within the cortex, they can generate potentials that are recorded from the scalp. Okay. Well, one of the problems that we face in EEG and ERP recordings is the volume conductance. So this is the biggest problem that we face in our field. And this is demonstrated by this figure on the right. You can see that a small dipole, a small electrical dipole, generate a potential that is widely distributed over the whole cortex. Moreover, you change the orientation of this dipole and you get absolutely different distribution, absolutely different maps. And because we don't know the real generators of the sources and their orientation, we have big differences in judging where are the generated of these ERPs potentials. So this problem is illustrated on this figure. I show you the electrodes that pick up electrical potentials from the scalp. I show the generators. Each generator, it produces a potential with some weight on the electrodes. And basically, we measured a sum of hidden, spatially distributed sources, and we don't know anything about the sources. And the question is how we can learn anything about the sources. And this is a formulation of blind source separation problem. So it's a mathematical formulation, but it's relatively simple, so I decided to show it to you. So basically what we record is on the left. On the left, X is potentials that we can measure. On the right, we see sources, different sources that contribute with some unknown weights to the potentials. Okay, so and the question is, if we know potentials, can we know anything about sources? And basically, we can formulate the mathematical problem. So we need to find out the mixing metric. And basically, this problem can be solved. So during the last years, we found quite many studies in the field of mathematics that they try to solve this problem, blind source separation problem. And there are many methods. In our system, which is called WinEEG, we are using two of those methods. One method is called independent component analysis, and independent component analysis assumes that the sources are stationary, stochastic, non-Gaussian processes with zero average. And the ICA assumes that these processes are mutually statistically independent. For example, any artifact like muscle artifact, like eye movement, different types of eye movement artifacts actually fit to this suggestion. So they indeed are stationary and stochastic, non-Gaussian processes. And the ICA can be easily implemented for these cases. That is why we are using ICA for extracting and for the correcting artifacts like eye movement artifacts and different types of muscle artifacts. So a good method, but you have to use it very carefully, because it's presumed that sources are stationary, stochastic, non-Gaussian processes. Okay, and here I show you the idea of blind source separation method, but I think that you can easily read it on the internet, so I'm going to skip this. The other method is a different one, and it assumes that the sources are non-stationary processes, but these non-stationary processes are not correlated across subjects. So if they are not correlated, so the cross-variance matrices should be diagonal. And this is a good method that can be applied for event-related potential. I think that you understand that event-related potential is actually non-stationary processes, and that is why you can hardly use ICA to those processes. The best way, of course, to use this joint diagonalization method. And we developed this method, and it's you can use this method by using a new version of WinEEG. Okay, so now we have methods. Then let us see how we can study different systems of the brain. Neuroscience tells us that there are several systems in the brain, like sensory system, like affective system, like memories, different types of memory system, motor system, cognitive control system. And these different systems, they involve different cortical and subcortical areas. And I think that you understand that to study this different system, you have to use different psychological tasks. Okay, so, and I'm going in this in this webinar, I'm going to talk to focus on cognitive control. What is the mechanism of cognitive control? What kind of psychological task we can design to study cognitive control? So what is cognitive control? So suppose you wake up in the morning and plan the day. First, you need to pick up your friend from the airport, and you ask your GPS system to suggest the possible routes. You select the routes that is appropriate to the context, so you are the time is pressing, so you select the fastest route, even it's if even it's include the pay route, the highway, the pay highway, and so you decide to take your car, start moving. The visual images may fit or may not fit your expectation. Then you see the accident in front of you. You decide to turn to find a different route. Finally, you are at the airport and you recognize your friend in the crowd. So you can see that this scenario that it's actually includes a lot of operations of cognitive control, like planning, selecting, inhibiting actions, detecting different types of conflict, and trying to take into account this conflict, inhibition of prepared action. So there are many operations in this scenario. And for studying this cognitive control operation, we designed the cued go, no go paradigm. And actually, in neuroscience, there are several paradigms that studied the cognitive control, and I'm not going to talk about all of them, but at least cued go, no go paradigm is able you to study not only reactive cognitive control, but also proactive cognitive control operations. Okay, I think that everyone knows who attended my courses, they know this task. The stimuli were selected from 3 categories, like animals, plants, and humans, with a sound. And the stimuli presenting in pairs, like animal, plant, and plant, human. And interstimulus interval within the pair is 1 second. And the intervals between trials is 3 seconds. So you can easily discriminate each trial. And the subject has to press a button to 2 animals. So when you see the first animal, you are preparing to receive the second one and to press a button as fast as possible. And when you see the animal, you activate the pressing action. You press the action. When you see the plant, you withhold from pressing. Okay, so, and you understand then in condition when you see a plant instead of animal, there is conflict, and the brain is detecting conflict, and the brain is suppressing a prepared action. Okay, so suppose you have a 19 channel or 32 channel EEG system, and you record event-related potentials in this paradigm, the cue go, no-go paradigm. So it's a referential montage. So EEG is a reference to linked years. And that's what you can get. So these are even the grand average event-related potentials, the ERP average for a group of healthy subject adults. And the green is go condition, animal. The red is no-go condition, animal, plant. And the black is ignore condition, plant. And this is FZ, CZ, and PZ central electrodes. Okay, on the right, you can see the maps of main waves in this task. So like C in V wave, negative C in V wave, like P3Q wave, so the parietal potentials generated in response to the first stimulus, to the cue. And then P3 go and P3 no go, and you can see that P3 no go has a more frontal distribution than the P3 go. It's those are well-known facts. There is nothing new, but what you see here is that the maps are overlapped. The maps are overlapped, and it's quite difficult to tell anything about operations, about hidden processes in this task. And the question is how you're going to solve this problem. Okay, again, volume conductance. And because of the volume conductance, the main waves are heavily overlapped. Okay, now I have to remind me about the Poisson equation that connects the potentials with the current source density. Okay, this is equation that we know from physics, and as you remember, I'm a physicist by education, and the Laplacian of the potential is proportional to the current source. Okay, so Laplacian, and Laplacian is a second derivative, spatial derivative is proportional to the current source. The question is how to do it. It's a quite sophisticated problem, and this problem has been solved. So one of the solutions is used in WinEEG, in the new version of WinEEG. So if you have the old version of WinEEG, then you have to go to MISAR website, and you should find the new version of WinEEG that enable you to compute the current source density montage. Okay, then now, so now we are using this current source density montage. So instead of referential montage, we're using current source density montage. You see the waves, on the left and the topographies on the right. So topography for P3Q, quite clear, you know, relatively local distribution. At PZ, quite clear the negative distribution of C in V wave. So it's a kind of a central distribution. Then P3 go, rather asymmetrical P3 go, and P3 no go. Again, quite clear the frontal distribution. You see, in this case, when you use a Caruso's density, it gives us a kind of a different picture. Here I show you the comparison of referential and Caruso's density montages, and you can see the difference. So basically, Caruso's density indeed gives you the information about the sources. Although still the question remains, what are the sources? It's quite clear that each distribution consists of several sources. And how to deal with this, and how to make sense of the component. Now we design 4 different tasks in which we try to actually to modulate different operations. One task is the basic task in which animal presenting, and they are the same animals. In the other task, the animals are different. I mean that the visual images are different, although they belong to the same category. In the third task, there are 2 different pairs, animal, the same, and animal, different. And in the 4th task, we ask the subject to press a button not to animal, but to animal plants. Okay, so we have these 4 different tasks, and we are now applying the methods of joint diagonalization of cross-variance matrices. And the first thing we do is we compute global field power. Global field power that was designed by a Swiss scientist, Dietrich Lehmann. And when you see on this picture, you quite clearly see 3 different stages of information processing. And roughly, very roughly, the first stage is fit to the phase of stimulus, visual stimulus detection. The second phase corresponds to recognition of the visual stimulus, and the third phase corresponds to action according to the visual stimulus presentation. Okay, if you go to the neuroscience and you look what is happening in the visual cortical areas, so then you can see a quite clear corresponding between what we see in event-related potentials, global field power, and in neuronal and intercortical responses. Okay, then this is what we have. Okay, so we have the first stimulus. We start with animals and we start with plants. And we can apply the method of blind source separation to this event-related potential. I have to mention you that in these 4 different tasks, we recorded ERP in quite a large group of subjects, healthy subjects of age from 18 to 45 years old. And we have about 300 healthy subjects, quite a lot of data. And using this blind source separation method, we selected these components. And then go and no-go trials, we selected this component, and the plant animal. We were able to separate this type of components. Okay, then I'm not going to talk about details how we analyze this data. It was a quite sophisticated procedure. And I'm going to, because I think that what you need to know about the component, their functional meaning, and how this component enable you to differentiate different brain dysfunctions. Okay, so the first component that we have here, we can select is a component which I call categorization component. So this is a map of this component. So basically generated in the occipital areas. As you can see, the green is animal response, and the red and the pink are response to plants. Then you can see quite clear that this is a component that indeed differentiate between two visual categories. This is the area that receives backward inputs from the amygdala. As you know, amygdala actually differentiate the emotional meaning of stimulus, and its main task of amygdala is actually to select the stimuli that they are dangerous for the human. And patients with anxiety, they have overactive amygdala. That is a well-known fact. But what is less known, that patient with anxiety has a much strong categorization component than healthy control. So basically, if we're talking about this component that generated in the occipital areas, we can say that this is a good indirect neuromarker of anxiety. So when I make analysis and I'm looking about, I'm looking at this component, and quite often increase of this component is associated with anxiety, high anxiety level of the subject, not necessarily having a diagnosis of anxieties, but having a kind of hidden anxiety. Okay, so the first component generated in the occipital areas that that differentiate visual categories and is associated with increase of anxiety. Is a good neuromarker of anxiety. Okay, that's what we know from neuroscience. We know that the visual information goes to the occipital areas, and then it goes to the areas of the temporal lobe, we call it the ventral visual stream, and then it goes to the dorsal visual stream. So these areas are associated with visual recognition. So damage to this area, to the ventral visual stream, they produces a visual agnosia. And this dorsal visual stream is responsible for association of the visual stimuli with different actions, because it's going to the premotor and some premotor motor areas. Okay, then the second component, as you can see from the map, it's a temporarily generated component. And this is a quite interesting component. And it looks like it has neurons that receive information from the top down visual areas and from the bottom up visual areas. So the information from the occipital cortex, bottom up information goes to the temporal cortex. The temporal cortex receives information from the prefrontal areas that keep information about context. And here these two pathways are united. And there are neurons that work like coincidence detectors. And they become active when there is a correspondence between bottom-up flow and top-down flow. And this area, they generate so-called N250 component, negative component, when the bottom-up and top-down operation, they fit each other. It's a good component. It's a good component, and it's impaired in cases of psychosis. So when the subject is in psychotic condition, and also if the subject is having autistic spectrum disorder, there is no correspondence between the top-down and bottom-up operations, and this this corresponds to the deviation of this component from the normals, from the healthy controls. So this is a good neuromarker of psychosis and also of high functioning autistic spectrum disorder. When I'm saying about this, I mean that we have quite large groups of patients with autistic spectrum disorder, with anxiety, with schizophrenia, OCD, etc., etc. So we also, as you understand, we have healthy control groups, and we have different types of patient groups. Okay, the other group is, the other component is a mismatch with working memory trace that is generated in the prefrontal areas, that is located in the inferior frontal areas. This is where the knowledge about the context and the high level presentation of visual stimulus is located. And if the stimulus does not fit this expectation, And then this area, they generate a negativity wave and 200 wave, which is which corresponds mismatch with the working memory trace. The next component that is generated in the parietal cortex, and they generated in the parietal cortex, and this component is impaired in a group of subjects who were premature born. So premature born subject, we have a large group of these subjects, and we compare them with the healthy control group. And we found the only difference in terms of event-related potential, the only difference between this group in this particular component. So this is a kind of P3Q component generated in the parietal area. And the functional meaning is association between the stimulus and action. Remember, when I give you instruction how to perform the task, and I tell you animal means pressing a button. And this is exactly pressing a button component, association between the stimulus and motor response. And is a good neuromarker of this kind of premature born subjects. The other component is a quite strong component generated in the frontal cortex. If you use the low electromagnetic tomography, Loretta or S-Loretta, then you can see that this component generated in the anterior cingulate cortex. This component, reflects conflict detection and monitoring. And it's impaired in a patient with obsessive compulsive disorder. In patients with obsessive compulsive disorder, after detection of conflict, so N2 component, then conflict monitoring, this is a P3 component, their brain generates a very strong N400 wave, which is not generated in healthy controls, and it looks like that the brain of OCD patient is constantly detect conflict, and this creates their problems. So this conflict detection is a good neuromarker of obsessive compulsive disorder, of obsessive compulsive trait. And the mechanism of cortical of function, the functional meaning, and of course the mechanism of generation of these waves are completely different, which is shown on this slide. I'm not going into detail, but believe me that although it looked like that this and the first component and the second component, they reflect mismatch, but they're using quite different processes, brain mechanism. Okay, then the other, you can see quite many components. We were able to illustrate quite many components, and each component has its own functional meaning and is impaired specifically in different brain disorders. And this component, which is actually impaired non-specifically, is impaired in all disorders that we studied. It's impaired in patients with ADHD, patient with schizophrenia, OCD, depression, etc., etc. So it's a kind of non-specific component, and its association with activation of the go and no-go programs. So remember that in the cingulate cortex, we store the goal, the drive of our behavior, kind of a model of behavior. And we also need to store somewhere in the premotor cortex the programs of this behavior. So the goal is decomposed into different programs. And in this case, in the go and no-go paradigm, we have two programs, go and no-go programs. And you can see that go and no-go program that generated different responses. And this is a good component for differentiating some psychiatric condition from the healthy control. So patients in all psychiatric conditions, they have impairment of this component. Non-specific impairment of this component. Okay, and finally, I think that this is the component. It's not finally, it's one more component that it generated, that had these types of map. And by looking at the event-related potential of this component, you can say that the go and no-go responses are reciprocal. That is exactly what we found in our studies with the basal ganglia. So if you put the electrodes in the thalamus, this is output of the basal ganglia, you will find neurons that had reciprocal responses to go and no-go condition. So basically, this component reflect output from the basal ganglia through the ventral lateral thalamus. And its associated with initiation suppression of prepared action via the basal ganglia pathways. And this component is actually impaired in patients with Parkinsonian disease. So it's a good neuromarker of Parkinsonian patients. Then the last component. This is a component generated somewhere in the supplementary motor cortex. That is the component that is associated with the general suppression of any action. And this component is a good measure, a good neuromarker of schizophrenia. Okay, and also it's impaired in patients with ADHD. Okay, so you can see that there are quite many components that can be extracted when you use current source density montages, and this method of blind source separation problem. Okay, so this is relatively new stuff, and I don't think that you can find this information in any of papers, because I just recently finished a study with a lot of subjects who are performing these 4 different tasks, and just recently finished analysis, and I decided to share this information with you and to demonstrate the beauty of this type of analysis and beauty of event-related potentials. Even though if you don't want to use current source density, if you are using the referential montage, still you can find this component. You can associate this component with impairment in some brain areas. Okay, so it is indeed event-related potentials indeed are a good tool for measuring activity associated with different brain operations, psychological operations. Okay, it looks that I finished my presentation. I think I spent 1 hour as we agreed of doing it. And I just would like to hear your questions.
Just going back through the slides.
Yeah.
Please explain.
Yeah. I'm all ears. Hello?
Thank you, Juri. That was a fantastic presentation and certainly a lot to digest. Some familiar material for people who've seen your sessions before, and some exciting new information that you've shared with us today. You commented that we won't find anything in the literature regarding this. Are there publications in the works that we can look out for?
Michelle, basically, this is the last part of my presentation is very new stuff. As I said, I just recently finished a study with 300 subjects, and all information that I showed to you that was done with this data. But also I was using some previous data that you can find in my last book, the 2016 book.
Thank you, Juri. So participants, you're welcome to sign off, but if you have questions, we have somebody there. Is it Kim? You're welcome to unmute yourself and ask your questions. So is it Kim?
Yes, thank you very much, Juri, for the introduction to the major components. I think it was a really wonderful review of major components. I just have one question regarding P2 component. You mentioned about bottom up and top down. Does it mean about the communication between limbic and subcortical and cortical area? Can I ask some clarification about that? Thank you.
Mm-hmm. Mm-hmm. Well, you know what? I think that if you want more details about this component, you can read the papers about phase repetition. There was a paper by some, I forgot their names, but a group of German scientists, they're presenting 2 phases. Okay, the phases could be the same or could be different, and they found that when the phases are the same, this area, they generate N250 component. I think it's called the N250 phase repetition component. but it turned out that it's not specific for faces. You can see.
Will be notified how to access the recording.
Sorry. So
That Romash Young, I don't know, some Hebrew name. 23 people from around the world. I don't know how they'd regard that, but that's a successful.
hello.
Okay. So the question was from Kim. I've just muted everybody else. Really
Yeah.
sorry.
Yeah.
So I think just to clarify, you're recommending for more information generally about P2 that there's research articles there. Kim, if I understood correctly, your question about clarification was around what did Juri mean by the top
Bottom
down,
up and top down.
bottom up?
Yes.
Well,
So by top down, bottom up, we're talking about the visual stream specifically, or we're talking about the thalamocortical or the limbic
Yes.
no,
reference?
I mean that we indeed are talking about the physical pathways. So there is a physical pathway that coming from the primary, secondary visual areas to the temporal areas, and there is a physical pathway coming from the prefrontal cortex to the temporal areas.
Yep.
Excellent. Thank you.
Thank you. Thank you.
Further questions, please.
And it looked like that there actually, if you are looking at the pyramidal cell in these temporal areas, so one input comes to the basal dendrite, the other input comes to the apical dendrites, and those neurons, they work as coincidence detectors. So when the two inputs they fit each other, the neurons they start firing coincidence detection. Of course, it's a kind of hypothesis, but we see it in ERP that indeed when there is a coincidence between two stimuli, this area generates quite clear and 250 millisecond component, negative component, which shows that it's associated with activation of some neurons. And I presume that these neurons are coincidence detectors. And as I said, there are several papers on repetition effect. This is a kind of a repetition effect. So when you present stimulus the first time and the same stimulus the second time, if they fit each other, then you will see this N200 component.
I have a question for Juri. This is Jen Markovics. It's a technical question. I see that your figures are of independent components from WinEEG, and it says below the topographical map. A variance. What does that variance mean? A percentage?
Oh, you mean this actually, as you know, when you get the components, you can hardly say about the amplitude, but you can compute the kind of the power of the component. So variance basically is the power of the component, how much these components contribute to ERP. So if the power, the variance is high, so the contribution to ERPs on the surface is quite high. If it's lower, then the contribution of this component to the ERPs is quite low. So the variance is the power of the component, the relative power of the component.
Thank you.
Thank you, Juri. There were a couple of other questions there. So if you haven't seen them in the group chat window, I'll just talk to those. Excuse me. One was asking about the recording. Yes, the session's being recorded, and in the very near future, you will receive an email to explain how you can access the recording for review at your leisure. And if you've got colleagues who missed the session, they're welcome to submit a late registration to Rustam so that they can access the recorded versions of the seminars. There was a question, Juri, about whether you've met with Bessel van der Kolk and if there are relationships between trauma history and ERPs.
Yes, that's I didn't mention about post-traumatic stress disorder, but we have a study together with together with the group from Sydney's Startis group, and we found one component that is critical for post-traumatic stress disorder. Yeah, there is indeed some specific component that is clearly impaired in post-traumatic stress disorder.
What component are we looking for this?
What component is impaired in post-traumatic stress disorder? The last component that is associated with suppression of actions.
Excellent, thank you. Then there was a question about are ERPs useful for clinical application to differentiate categories of subjects?
Well, as I tried to show, indeed, the ERP can be used for this, and during the other webinars that we schedule, I'm going to talk about application for ADHD, application for schizophrenia, for different types of disorders. Yes, and what I'm going to do is I'm going just to find some case of patients with, say, ADHD or schizophrenia, whatever, and to analyze it and to show how we're going to use this data, ERP data, to make diagnosis and also not diagnosis but prognosis, what to do or how to treat these patients.
That's excellent.
This is the most important thing.
And
so
I think it's exciting given that there's so much, well, let's say that the utility of the DSM has been questioned over and over again, DSM and ICD, based on, diagnosis based on externalizing behaviors, and the RDoC approach is having us really look at the brain bases, the neurological function, as opposed to the externalizing behaviors. And this is a really exciting space for ERPs, looking at the RDoC research as well. Juri, there's another, sorry, you wanted to comment on that?
No, I completely agree. The ERPs, they provide you a unique opportunity to go deep into the brain and to study the brain correlates of disorders. I completely agree.
Especially when we're looking at biological treatments, we want biological data. So yeah, really exciting work. Another question, looking at, would we see the same components when using other methods of processing, such as reference to CZ or average?
Now, I think it's a good question. And I think that, of course, it's a good idea to do the current source density montage. But if you don't do it, and if you apply the blind source separation method for referential montage, you will get the same component. So it's actually, it is not critical using current source density montage. You can do it.
Other than linked years, if you're using a CZ or an average.
Yeah.
Similarly, okay, brilliant.
I did not do anything with CZ montage. I don't know, but it looked like yes. And I did it with a common average montage, with referential montage, with the current source density. You basically get a similar result.
Thank you. And just going back a moment to the question about ERPs and PTSD, a couple of people have commented in the chat window, so have a look there. Jen Markovic's commented that in the P3A novelty component is increased in PTSD, and Darius has provided us, thank you Darius, with some web links to the research on this topic that's been published, and some information about that from the Trauma Center in Sydney.
Oh, very well, very well.
All right.
Very, very good. Okay.
All these components are really new. Yes. Okay. Perhaps this will be teased out in subsequent seminars over the next number of weeks, but Silvana asked, this stuff is difficult to understand and follow. Yes, we all agree. Is it possible for Juri to give correlation of the between the new ERPs that you're identifying and the old components?
Yeah. There is quite clear relationship. Yeah.
So you're able to include that in the next seminar just to help us see the differences?
Yeah, I think so.
Thank you so much.
So basically, what I try to do, I just try to show you the new stuff, and I wanted to give you impression that we know quite a lot about functional meaning of the component. But basically, you're absolutely right. You can use different montages, and you will get similar results, similar components.
Okay.
Absolutely.
And if your intention was to give us impression, just on behalf of the crew, we are impressed.
That was my goal, to give you the idea that we can derive from event-related potential from all this method that we are using quite a lot of information, very detailed information. So I tried to show you that we are not confined by P3A, P3B component. It's much more sophisticated, and there are details that you can look at using the event-related potentials. Yeah.
Thank you, Juri. And the final comment or request here relates to the specific conditions in which we might look at ERPs. And for those of you that don't know, after this introductory session, we have a subsequent series of 6 topics looking at ERPs specific to ADHD, ASD, schizophrenia, OCD, Alzheimer's disease, and then in peak performance. But, you know, because we know you've got plenty of time on your hands, there's a request to add another topic on depression. Is this something in which you have ERP evidence?
Yeah, we have some data on depression. Yeah, I can actually do it.
So perhaps we could add that one for later in January.
We will see. Maybe when I will talk about, say, maybe we will combine with the depression with some other, like OCD webinar. I don't think that it's wise to do it a separate webinar on depression.
Okay.
We need to combine it.
Okay.
We also discussed doing webinar on PTSD, and I think Juri was positive about that.
Yeah. Okay, well, we're getting lots of thank yous as people start to sign off. Well done, Jen, and others who are working through the middle of the night to participate in this session. Thank you again so much, Juri. We bid you farewell.
One more comment.
Please.
So people are welcome to send me the data. So if they want me to analyze their patients, they have difficulty of analyzing. So please, I think that if any of you is using this go, no, go paradigm, you can send me the data. and I will analyze it, and we'll work how to extract this component in individual subject, how to compare it with the normative database, and how to make conclusions.
So what we might do to facilitate that, Juri, is I'll talk to Rustam, we'll set up a Dropbox, and when we send out the link to this session recording, we'll provide a Dropbox link so that if anyone has data that they would like to have analyzed in one of the subsequent sessions, please de-identify the data and provide a little, little context, but upload it to the Dropbox, and then it'll make it easier for Juri to access from one space. So we will organize that as soon as possible. And similarly, if there are other questions or topics you want to address, just drop them into a Word document and upload them into that Dropbox. That way, all of the incoming and outgoing can be in one organized space. Thank you, everybody.
Yeah, and one more comment. I understand that it was difficult for you to digest, and this is relatively new stuff, and there are much more that is hidden behind what I presented. But you know, we have some webinar in front of us, so we can discuss all details later.
Yeah, Juri, I have one final request. I've encouraged people to upload information or questions via the Dropbox that we're going to provide them. But there is a question or a request about receiving your email for direct communication. Are you willing for that, or would you prefer just, that's okay?
Yeah,
So which is the email address that you would prefer participants to contact you?
I mean, I have only one email address, which is Juri Kropotov at yahoo.com.
All right, I'll just pop that into the chat window for everybody to access. Thank you, Juri.
Yeah, you're most welcome.